So we've been looking a lot at how plants learn to recognize themselves and each other, kin versus non-kin. Um, and then this paper was looking at, can plants recognize that there's another plant near them? And if there is a soil system that they're growing in that has patches of better patches for food, like minerals, like this is a really good patch for minerals. This is not so good or more dilute. How do they behave when they are faced with some options? So Cahill, James Cahill is the senior author on this, and he's done a lot of work with looking at behavioral ecology of plants. And in fact, I just posted a pretty short paper. It's like a perspective or opinion paper on his ideas that we should be doing a whole lot more of looking at plants through the lens of behavioral ecology. We do a lot of behavioral ecology when we look at animals and how they are behaving and interacting, but he says it's time to do that now with plants. So this is one of the papers that he's published looking at this idea. It's a pretty big paper. I'm not assigning it for reading. I'm going to pick out some of the, I guess, main points that he was making through this research. So this was kind of like the part of the experimental design. He did many, many experiments, and this was where he had sunflower. He was working with sunflower plants. And so these are like we call them mesocosms, but essentially like a growing area for the plant. And you would have strips of soil that were really high, like if it's dark gray like this, that means really good mineral nutrition, like nice dense options for nutrients in that, whereas the one over here on the left, not so much. And so they would grow sunflowers and say, you know, if you're a sunflower and you're growing in a situation where you have lots of nutrients in that gray area of the soil and modest, which way are you going to grow? You're going to go toward the right in this case, right? You're going to go after the nutrients that are in that high nutrient patch. So they would test sunflowers and say, how do you behave when you're all alone and you're given these options? How do you behave in the middle one? How do you behave if you have a neighboring plant growing and on either side of a good nutrient patch? What are you going to do? And so in this, what they were focusing on is this plant right here in this middle picture. They're saying, what does that plant do when it has a neighbor that's going to grow next to it and it has the option of growing toward the high nutrient patch, but then it would be in potentially direct competition with the plant on the right. Or it could grow over to where it's more modest nutrient and not directly compete. How is it going to work? So they set that one up. And then this third one on the far right, there have two plants. This is the one they were zooming in on and took measurements off of that one. They said, what's going to happen when it has that nutrient patch right to the right of it is better than the one to the left. And it also has a neighbor plant to deal with. So here, the next slide shows some of the data. All right, so 1P alone, so that means if you just have this one plant, this one sunflower plant, and the yellow outline to the right, that's the better nutrient patch. And then they were measuring root length on the graph here, graphing it out over as it grew from day 0 to day 16. And you can see, well, what does it look like it's doing? Is it favoring growing toward the nutrient patch or to the kind of wimpy nutrient patch. They have it color coded. Can you see that? And so they're saying there's more root growth into the nutrient patch. When it's all by itself, given the choice of on the left, I have an okay nutrient patch. On the right, I have a really good nutrient patch. I'm going to grow into the high nutrient patch. And that's what they show. Then they say, okay, what if there's a neighbor plant? And so they were showing that when you have a neighbor plant into the mix now, you would think, this plant is what they were, the left plant. They're saying, what's it going to do? You would think it might want to grow toward the high nutrient patch off to the right in this case. But they find with the data, it was kind of even Steven. You can see that these two are kind of lining up. There's no obvious difference. So that plant was growing roots out to the left and to the right. And the only difference is that there's a neighbor plant next to it. And you can clearly see that it changed its behavior. Instead of doing this, like what you see on the control on the far left, you have a much different growth pattern just because there's another plant in the mix. Then they said, okay, what about when you have one of them closer to the good nutrient patch, the high nutrient patch? Ugh. And you can see that, again, you get that really great root growth into the high nutrient patch. It did not go into this other nutrient patch where it was closer to that neighboring plant. So when a high nutrient patch was equidistant to a neighboring plant, they avoided. They're like, oh, okay, I gotta, do a little bit different growth pattern. And like I said, the only difference was that there was a neighbor in the mix on that one. And then with 2C, when the focal plant was closer to the good nutrient patch and the neighbor plant was farther, the focal plants did go toward the high nutrient patch. So what these studies were, let me go into one other result that they were doing. Here's another one where in this case, they have two high nutrient patches set up. Okay, so the dark gray columns are high nutrient. And you're seeing that if that sunflower is growing all by itself, it will kind of even Steven go to the two nutrient patches. And so everything's kind of the same here in terms of their work growth. But look at with the figure B there, figure 3B. Now the only difference is they put a neighbor in there. And then they asked, how is that plant going to behave? So they're zooming into the right plant and saying, how is that plant going to behave now that it has a neighbor? And you can see that it preferentially shifted its growth off to the nutrient patch to the right. So in this study, when they did the calculation, the plant ultimately invested 40% less root length in the patch that a neighbor shared equal access to. Right, so big difference. That's a big difference in terms of how it was deciding to shift its root growth. And so really the only difference here when you look at the control versus, the test example there, the treatment, they just added in a neighbor and said, how is it behaving when you have that situation? And so, yeah, with this case, they were working with the sunflowers. They said, yeah, they behave differently if they were growing all by themselves versus if they could sense a neighbor. And spatial social context, that's how he refers to it. It's a social context. Am I alone or do I have a neighbor? And that does make an impact on how the plant decides to go ahead and grow its roots toward different nutrient areas in the soil. So they integrated spatially disparate cues. So they're looking at a neighbor plus a good nutrient patch. Those are the disparate cues. Now I have a neighbor. Oh, here's a good nutrient patch. And those were both influencing what was going to happen in terms of their root growth. Plants can use social information in their foraging decisions. So they use information about the specific spatial social context to inform their responses to high nutrient patches. So this is showing that plants use information about their social environment to modify root foraging behavior. And so this is also kind of backing up, the idea of kin versus non-kin, how kin plants tend to not compete so much with each other in terms of their root growth. They're more restrained. And so this is something that is adding to this piece of evidence that you have their ability to not only sense in the soil where they should grow in terms of good nutrients, but they're also sensing if they have neighbors or not. And based on that idea of, do I have neighbors or not? That might shift how they grow into the soil. And so then he published this paper. That's the one I posted, the inevitability of plant behavior. So it's just a short read. It's just kind of like an opinion piece where he is stating that, we really do need to look at behavioral ecology when we look at plants. The evidence is mounting that it's clear that they are social organisms and they use that information in order to survive and get their needs met in complex environments. So give that a read, see what you think. See if you think he made a good argument or not. But that's in the EDUCAT under the readings and videos. So he was also one of the people interviewed in that What Plants Talk About video. He was the one that kind of kicked off the initial part of the start of the documentary. So very much into behavioral ecology, and he's very much into root foraging and showing how they definitely have a way to go and forage their roots. I think that he made the link in that video of bears, like a mama bear with the baby bear going into a patch of blackberry bushes and hunkering down and eating them all up. And then he showed the roots growing into a nutrient patch and just staying there and growing more roots and just plunking down and eating that all up. So a nice parallel there between bear behavior and plant behavior in terms of foraging for food. Okay, so that's the end of that long slide set. I see I'm at 102 slides. I think we should stop there. Yikes. Um, Even though I could go on and on and on, but we shall move on. All right, that's not quite what I wanted. So then, okay, the next thing I wanted to start in on. Well, actually, we're going to revisit some ideas in this next topic, but apply them to a new topic, kind of. This is the next slide set. Managing plant microbe interactions in agricultural with that kind of slant. Okay. So the next, I guess, section of this class, we're going to look at what are they thinking about in terms of agriculture in this day and age, we're going to think about it in terms of what are the hot topics. We'll get into some of the really cool cutting edge discoveries and some of the companies on how they're doing research and formulating products that can be put to use in agricultural right now. We'll look at some ideas of the future, like what's being worked on but not quite there yet. And I'm very happy to see that one of the big pushes right now in agriculture is we can't ignore the microbes that coexist with the plants. I mean, that was kind of the whole part of when I went to grad school. That was my focus. And no, it seemed like hardly anyone was doing it back then because they had back then, way back when I was in grad school, we had all these herbicides and pesticides and fungicides, and farmers just relied on those really heavily. We'll just spray that onto the fields. That'll take care of this pathogen, that pathogen. Well, then there came the idea that, maybe these chemicals aren't so great for us, and maybe we should consider doing something else. But the farmers really wouldn't do that until the government started phasing out pesticides and fungicides and saying we're going to ban them. Then that's when I could see a real big push for, uh-oh, we need to find something else to do. We can't just rely on these chemicals. We need to figure out other ways. So that's when they started getting into integrated pest management strategies where you attack the issue from many different angles and put together. Hopefully that will do the job. And then as part of that, the microbiome came into more heavy scrutiny. So this is the idea, again, going back to the holobiont idea, where you cannot ignore the microbes that coexist with the plants, whether they're the microbes inside the plant as endophytes, or on the roots, or in the soil right next to the roots, on the leaves, and so on. So this is a figure taken from a paper that is looking at this idea and how we can use the microbiome to help the plant. One of the ideas that I think you probably learn in a basic ecology class is that diversity. When you look at a certain habitat, if it's really diverse in terms of the number of species there, that usually means a relatively healthy ecosystem. Greater diversity is a good thing. And then they're finding that with the human microbiome too, that if you have a nice, diverse human microbiome, you tend to have healthier hosts, healthier humans. So we'll think about that a little bit. This dysbiosis idea, that's if the microbes get out of balance from normal. And so if they move away from a healthy state in terms of the microbe, either the numbers of microbes might crash or the ratio of who's there might really shift quite a bit. That can cause what they call dysbiosis. You have all kinds of microbes that can work together. So that synergy idea that when they work together, they actually do a much higher positive thing than if they worked alone. And then, of course, you have antagonistic interactions. We got a whole lot going on. It's very complicated, but the title that's the title of the paper. Plant microbial diversity is suggested as the key to future biocontrol and health trends in the agricultural system, primarily, is what they're focusing on. Previously this semester, we looked at the idea that plants are not just sitting ducks, sitting in the growing in the soil, and just everything happens to them and they have no choice. They have a lot of choice by how they interact with their environment in terms of they can recruit certain microbes to them. They can inhibit microbes from being around them. So it's very targeted. That's the word they like to use. It's a very targeted thing that plants will do to their surrounding microbes. And they release a bunch of different chemicals and organic acids, and I mean, on and on and on. Lots and lots of different chemicals and nutrients that get released by the roots on a pretty continual basis. including secondary metabolites that they're making and specifically making for the purpose to excrete them out to recruit certain microbes. So it's a very targeted thing. If you're going to make a secondary metabolite, that's effort on the plant's behalf. That's using up resources, so you better do it for a good reason. So they're excreting all kinds of chemicals that can attract or repel. They're turning on signaling pathways to get this done. So again, it's an investment in resources and carbon and other nutrients for them to do this. But it allows them to be involved in their habitat in the sense of they can recruit or inhibit. We know from previous weeks of this class that microbes also are impacting plants. This is just a schematic showing that they're everywhere. They're all parts of the body of the plant. You can find microbes. And of course, in the soil right next to them. And they're doing all kinds of stuff. They are impacting growth all the way to the seed germination stage onward. They're helping the plant get access to nutrients, especially some of the big ones, nitrogen, iron, phosphorus, potassium. Of course, the nitrogen fixers, we talked about those. Those are very special microbes. They're helping defend against plant pathogens, helping them deal with just everyday stress. Maybe it's a drought period or it's getting really cold. And they can influence definitely what the plant is doing in terms of secondary metabolite production. You can see that the presence of the microbes do influence that all the way from, how certain they're looking at, the essential oils found in basil, for example, or the THC levels or CBD levels, the presence of microbes makes a difference. And so there's all kinds of, and the ethnobotany is really starting to look at that more, like how do the microbes influence the certain oils or the certain chemicals that we want when we're using plants in that ethnobotany realm. I think I showed you this before with the plant growth promoting rhizobacteria, just trying to show again the complexity, lots of things going on in the soil system, on the roots, in the roots. And of course, the plant is doing its part as well. Seems like everywhere they look, they find new ways in which plant microbes are affecting plants. They even are showing differences on how insects interact with plants, depending on the presence or absence of certain microbes. And it doesn't have to be like, these are typically soil microbes where the insects are like on the shoot or leaves. And it's actually the microbes in the soil, the presence or absence of those is making a difference. So it's not like the microbe has to be right next to the insect for this to happen. They're looking at pollination as well, that microbes, especially some of the endophytes, are influencing chemicals found in flowers and nectar. Phenology is just looking at when does a plant decide to flower or when does it decide to drop leaves. You're kind of looking at key parts of its life cycle. That's when they talk about phenology. So we can see that happening. If, of course, we've talked about the idea that they are a type of epigenetic mechanism on the plant. And of course, we even touched on the idea that they now are pretty certain that mycorrhizal fungi were instrumental for getting the plants to be able to handle a terrestrial environment. Go from the ocean onto land is a big, big step. So we've gone through this, a lot of this you've seen. But now let's think about how are they considering this interaction and applying it more toward agriculture. This is a published paper where they're proposing we need to look to nature to figure out how we can do it better in agriculture. And so this was the idea of, okay, well, if you have a healthy ecosystem, you tend to have high diversity of plants. They're trying to show that on the left here, where you have lots of different plants And all the different colored symbols here are the diverse microbes that are showing up because of this very diverse area of organisms. They're saying your root systems, so the differential display of roots. That's kind of getting at niche differentiation, that they're able to share that soil resource where they're not all competing for the exact same patch of soil. They have different areas that they're tapping into. You have lots of different exudates because there's different species. Okay, and so this is some of the characteristics that they point to for a healthy, forested ecosystem in this case. So they say, can we take this and these ideas and try to create that in an ag system? What do you see in that agricultural field? What did they draw there? Can you tell? Corn. Do you see any other plants there? No, it's just corn, 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 corn. So conventional. So when you say conventional agriculture, you're typically talking about that idea where it's a monoculture system, high input with chemicals and synthetic um, fertilizers as well. And as as a result of that, they're trying to show that there's less diversity in microbes, and that if it's just corn, you're probably not getting a lot of diversity in what they're excreting from their roots. So there's a lot of differences between a healthy natural ecosystem and an ag field in the conventional agricultural system. That's changing in the sense that there's more farmers that are going into organic farming and doing like permaculture farming, that kind of idea. And we'll talk a little bit about that later, but so there are different ways of doing it besides just monocultures. So this title is kind of odd, but microbe assisted crop improvement, a sustainable weapon. I don't know why they chose weapon. I would have said a sustainable tool to restore holobiont functionality and resilience. Doesn't that kind of give it a negative tone? Sustainable weapon. I don't know. Maybe it's just me. But this is the abstract looking at the idea of when you breed corn plants for production purposes, you're really, humans are intervening, right? You're doing selective breeding. So, and it's, traditionally done through human intervention of you keep breeding crops and looking for the next generation, the traits that you want, and then keep, honing in on the traits that you want. If you're a corn grower, what you're really often going for is really good cobs, right, with really sweet corn kernels that will give you, when it gets to market, you'll have a good return on investment. Well, as you're breeding for that, other traits of the plant are not often thought about and go by the wayside. And so the point, and we'll talk a little bit more about that, but basically they're seeing that because of that, I mean, during all those breeding processes, they didn't really think about the microbes that hang out with these plants and what's happening to that interaction. And so they're saying, our anthropocentric point of view has really created some issues. We had a lot of corn plants that are, same genotype. So you get loss of genetic diversity. You get really shifted plant microbe interactions through all this selective breeding. And so they're saying, you know what, we could try to help this situation out by really turning to the microbes and saying, can we understand them better? And then can we use them to improve some of these plants that have been highly changed from their wild relatives? And some of the traits are definitely missing because of all that selective breeding. So they actually call it domestication syndrome. That's the term that they use when thinking about this idea that if you're intervening and doing selective breeding on those plants, it's gotten to the point where a lot of these plants can't survive on their own without human help, either through, intensively irrigating the plants, applying pesticides of some sort, and just all the aspects that you think of when farmers are growing corn, for example. You can't just plant the seeds and walk away. You have to intensively farm throughout the growing season. So that's a lot of fertilizers that get put in, a lot of pesticides that get put in, and the resulting often environmental damage that can happen because of it. You also get that reduction in genetic diversity through all that selective breeding. And that tends to allow for pathogens to, it's kind of a weak link. So pathogens have a little bit easier time of getting in there and causing problems, especially with monocultures. Once they get into that field, it can be devastating. And I think I might have mentioned this before, but I'll say it again with, even with the fertilizers like the NPK that they add into the soil. And then you think about all the different pesticides, fungicides, herbicides, all of those, but even the fertilizers can damage the microbes that are in the soil, can kill them. And what they end up seeing is that when you have a conventional ag field that's been in production for a long time, they actually refer to the soil system as a microbial desert, a desert, because there's so few microbes compared to control soils that haven't been intensively managed and used for crop production. And so even fertilizers can kill microbes, even though those are nutrients, but they can still cause issues, as well as some of the nasty chemicals. So it's really created a situation where these monoculture fields don't have a lot of the microbes they once had. So that's a big deal right there. And so they're starting to realize that, all the selective breeding has really created a lot of unintended consequences. And now with the big push of, we have so many more, what is it, by the year 2050, what are they saying, 10 billion people on this planet, I think they're saying. It's, if you go to conferences where you talk to people who, are in the agricultural realm, they're a bit nervous. How are we going to do this? It's not, you walk out of those seminars going, ooh, I need a beer. So it's kind of depressing. But luckily, there's a lot of options that are in the mix that are going to work, I think. So, but it's still, we have to do better. And so this is where they're doing a lot of comparisons back to the wild relatives. Luckily, we still have a lot of the wild relatives for some of our crops, and they've been able to save seeds from those wild relatives. And so there's been some nice comparison studies, and they're trying to figure out what can we do to put a little bit more wildness back into our heavily tended plants, like corn, for example. So they are starting to look back to the wild relatives for that. This is a study that they were doing and kind of using the idea of grapes and grapevines and trying to show, this is a visual, just trying to show some ideas here of what could we do to try to improve this. So on the left, you have kind of a sad looking grapevine where the growth isn't great, not many grapes showing up. So they're showing growth down, but they're defending themselves really well. So they're talking about this trade-off. This is a real thing. All right. So if you're going to defend yourself, you got to stop growing. You can't do it all. And so they tend to see if you're getting attacked and you have to defend yourself, your resources are going toward that. You're trying to stay alive from this, whatever predator it is. So that means you slow your growth down. And so on the left, that's what they're trying to show. And then on the right, they're saying if you put all your energy into growth and not into defense, then these red targets are supposed to be, you get attacked. That's what those red targets are supposed to be. All right. You put a target on your back, so to speak, because you're not really into defending yourself. You're just into growing. So that's the scion, by the way, if you've not heard that word, that's the top part. When you do grafting of grapevines, the top part is called the scion, and the bottom part that you graft onto is called the rootstock. And so with grapevine production, you do this grafting onto rootstock, and they're kind of showing the same thing here, where if you're defending yourself, you're not going to put a lot of effort into root growth. But if you're busy growing, then you're not going to put much into defense. And so that we always talk about this trade-off. How are plants dealing with that? How can we help plants do this better? So this term, SynCom, is apparently coming into the mainstream. I first heard that a few years ago, and now it seems like it's more commonly showing up in the literature. SynCom stands for synthetic communities, in the sense that the human is choosing. I want this mixture, this cocktail of microbes. So I'm creating my own community. It's very human created, so they're calling it synthetic communities. And so they're saying if we can kind of like with the when with AIDS disease, they create a cocktail of different drugs, and they don't just use one drug to try to get that under control. They use a suite of different cocktails, and so that's the general thought with microbes. You tend to have to do a mixture of diverse microbes, so that when you add those into the soil, generally a high diverse set of microbes should be better than if you just have one type. And so they're trying to show that by all the different drawings of the different types of microbes. And they're saying if we can do this in the right way, of course, the ultimate goal is let's get really good growth. So lots of grapes in this case. And we have a more balanced idea where the plant can grow, but because of all of the helpful microbes, they have that extra help for defense. And so they should be able to balance this better in terms of their overall growth is good. They don't have to defend them. They don't have to put as much resources into defending themselves if they've got some help. Does that make sense? Okay. So microbes, and it's a great idea. And this is definitely a lot of scientists are working on this. You have to know a lot about your plant and your soil system, and you have to do a lot of research on the microbes in order for this to start paying off, but it's doable. Okay, so a lot of what they're doing is looking at the soil microbes, and more people are looking at endophytes as well. So this idea has been coming about for a while now. This paper was published in 2016. Back then it was called soil memory. It's kind of gone by the wayside now, but I like it. I wish it would have stuck. Now they're calling it plant soil feedbacks, which to me is more vague. But anyway, it's plant soil feedbacks, and then they'll label it as PSF. So you'll see that in maybe some of the, whoops, PSF, plant soil feedbacks. They don't even mention microbes in that term. It's like, no, it's microbes, not soil. But anyway, soil memory as a potential mechanism for encouraging sustainable plant health and productivity. So back in 2016, this is Jorge Vivanco's group. He's out in Colorado. He does a lot of work with this kind of idea. And so these are some quotes taken from that paper. They're saying, if you look at the microbial communities that are in the soil and study them over time, over multiple crop cycles, over time, they seem to have some of the same properties as what we see when you think about like the human microbiome, and like when a baby is born, that the baby will get colonized by primarily first by the mom, and all the microbes get transferred from the mom, but then you get other family members, and they're saying that this mixture of the microbes in the rhizosphere is very similar to what's going on as the human microbiome is developing from childhood into adulthood. And so they're making that parallel thought between those two. And so they're saying that it has like a soil memory that can get passed to the seeds that land into that patch and start growing. The previous plant that was there is leaving a soil memory of microbes in that soil system. And so they were using the term soil memory because when plants are growing in the soil, they impact the microbes and are changing the types of microbes that are there, the ratios, population levels. And so you'll also see this term, soil legacy, the microbial soil legacy, persistence of plant-mediated microbial soil legacy effects in the soil and inside the roots. So this one was just a few years ago. This is a 2021 paper. So that's the plant soil feedback now. That's starting to come, that term is starting to be more common in the more recent papers. So depending on what system you're looking at, you can definitely have situations, that second bullet point, you can have pathogens building up in your soil. So if you think about when I was a postdoc, there was a field station where they were growing different crops and studying disease. It was a plant pathology department. And one of the fields that we got involved in was a potato field where they could grow potatoes in that field because they knew there was high levels of the pathogen in there. And they wanted that because then they wanted to put in different cultivars of potato to see if it could handle being in diseased soil. Which one could we breed for resistance? If we put these different varieties of potato in there, watch to see how they handle all this pathogen. Maybe we'll hit on one that is resistant, and then we can breed that one and have a resistant potato. It was called potato scab disease. And so they had been doing that for decades. And that field was a scabby field, lots of potato scab disease. And then kind of slowly over time, the they dig up all the cultivars, all the potatoes at the end of the growing season, and over time there's less and less disease showing up on the tubers. They're like, what is going on here? All the cultivars had less disease. And then when they took some soil samples through the field, they realized the pathogen was disappearing. There was hardly any pathogen left. And so those plant breeders were like, well, that stinks. We need a field that has really good scab disease. That's how we test our cultivars. So they abandoned that field. And that's when my postdoc mentor said, well, what happened to that field? We have all, good disease for years and years and years, and not all of a sudden, it took a while until there's virtually no disease. So what happened to create that situation? So that's when she went in and said, let's study that soil system. Let's figure out why there's no disease in there. And so by them planting, the reason that field was so high in disease originally was that it was potato, 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 potato for years and years. And so the pathogen built up in that soil. It's just that after a while it went away, and I'll talk about that as well a little bit later. So you can have that situation, or you can have the opposite, where you get a lot of really good microbes hanging out in that system, and it's a very positive thing. And it depends on the plant, all the way down to the age of that plant, the species of the plant, what are they doing? And so it's plant dependent. It's going to require a lot of research. The one thing that's starting to become clear is that young plants, seedlings, so when a seed first germinates and starts to grow, they seem to be more susceptible to getting endophytes in. Like they're more receptive of it's time to get some endophytes into their body. And so that's an interesting thing that is pretty new. That's not something that I've seen in the literature. This is actually the first report I've seen of that in this 2021 paper. And they say maybe that's because younger plants, like kids, their immune system isn't quite up to snuff yet when you're a little kid. It's kind of that thing, like, when you're really young plants, maybe your defenses aren't all up and running to full potential. So having some nice endophytes in your body might be a good thing. Okay, so they're looking at these ideas like how do the microbes respond to plants growing in there? How do they persist through multiple generation of plants? So this idea that when you see it, when you look at a seed, you have to imagine that inside that seed, there are endophytes in there that came from the mother plant. But now they're saying that, all plants will select endophytes from their habitat. Once that seed germinates and grows, they will get more endophytes through that process as well. And so you can get endophytes that vary. There's endophytes that came with the seed, plus then there's fresh endophytes that get taken up when that seed germinates in that soil system. So they're looking at this idea. If there was, previous plants in that soil system, what kind of legacy did they leave in terms of the microbial composition? Because those plants did impact the soil microbiome in some way. And so they're saying, you have that whole situation to think about. Are different plants leaving different soil legacies? Is there any kind of way we can look at this and use this information when we think about agricultural growth production? So that's what some of this study was looking at. This is actually, I think this study was done I can't remember where this was. Oh, in the Netherlands, I believe. So here is a mock-up of what they were doing. This is what they're doing in this paper. I've read many other papers over this past couple of weeks that are doing very similar ways of running these experiments where you have to invest some time in this because in this particular case, they grew plants for 1 year in soil systems, say monocultures for 12 months. So that's your previous plant. Then they took 4 different species and put them into those previous plants' soils. Does that make sense? So when they had that 12 months, when they did this step, they call it conditioning the soil. Those plants grew for 12 months in that soil. They're busy excreting a bunch of stuff into the soil. They're conditioning the soil. They're impacting the soil. The microbes are impacted. Then they say, okay, let's get rid of those plants, keep the soil, get rid of those plants and start putting in different plants. And they did it in every pairwise, every combination. And then they said, we're going to grow those second plants now and take some sampling. I think they did 1 month, after 1 month of growth, 3 months of growth, and 5 months of growth. Okay, so 1, 3, and 5. And then at the very end, they destructively sampled for endophytes, but they were taking soil samples throughout and plant measurement. And at the end, they destructively sampled. So that's kind of the general process that they use when they do these types of studies. They condition the soil and then they grow another set of plants in that soil and look to see what happens. Okay, we'll stop there. We'll get into the results after break because it's already, so we'll come back at like 3:02-ish, according to my clock. Did here on the experiment. Okay, so they're looking at what's happening in the soil, what's going on with endophytes. So when they were collecting their data and looking at it, they did see a soil legacy. So the previous plants that had been grown from a year in those pots of soil, and they saw that the fungal community. What happened there? Okay. They could detect a legacy for at least, so they were testing at 1 month, 3 month, 5 months, taking samples. And they said all the way out to 5 months that they could see the soil fungal community there was a legacy in that group. And then the effects of the previous plant on the soil community outweighed the effect of the current plant. And so they're saying with the fungi, it was the plants that were grown for a whole year in there that seemed to really impact. And when they popped in those new plants for 1, 3, or 5 months, well, they did it for a full 5 months. The fungi didn't shift that much. Then with the bacteria, they did both with the endophyte community. Again, the previous plant seemed to, so that soil legacy from the previous plants that were in those soils for that year definitely was influencing the endophyte community of the current plant that was in there. Where was the other point? There's another point in here. Where did it go? Oh, here it is. Soil bacteria was a little different. Soil bacteria, the plant that got planted into the conditioned soil seemed to really modify the bacterial composition, and so the legacy wasn't as strong when you looked at the bacteria in the soil. And so it seems like the fungi stick around longer. They're really impacted from those 12 months of growing with the previous plant. When you pop a new plant in there, the fungi kind of stayed strong, whereas the bacterial community started to shift in response to the current plant. That's one of the points I'm trying to make here. And then with the endophytes, it seems the previous plant, what was going on with the microbial composition after that 12 months of growing was really influencing. And so you can imagine when they popped the seeds into that conditioned soil and the seeds germinated and grew, those plants were starting to take up endophytes at the seedling stage. And so it kind of makes sense that the previous plant's legacy was really the strong impact there, because they were immediately taking things up right from this, germinating and growing as a young seedling. Okay, so there's lots of studies coming out now on this idea of soil legacies, because when you think about agricultural fields, sometimes it depends on how they run it. Sometimes a cornfield is always a cornfield, but sometimes they rotate and do other plants through that field over time. And so they have to think about these ideas of soil legacy and how that would impact the growth of the plant that comes next into that soil system. Another idea that they're looking at is soil immune responses, kind of an odd phrase, but thinking about disease now, when disease comes into a soil system and starts attacking plants, does the soil reflect some kind of immune memory of that disease? Is kind of an interesting idea. And so, this is again being applied toward agricultural systems. They're saying that we have to pay attention to the soil microbes in the agricultural soil. I mean, without plants, we'd soon perish. So we need them for everything. And so they're like, okay, let's look at soil microbes, and can we create a way to get the soil systems to be immune, so to speak, from the pathogen coming back into that system. So this is the idea of kind of what we were studying or kind of happened upon with that potato scab suppressive soil. So those are actual terms that they use in this realm of study in plant pathology. So conducive, if the soil is conducive, that means it's got a lot of the pathogen present. If you plant plants in there, they'll get diseased. It's a conducive soil. So they're trying to show a snapshot of a picture of a plant, several plants in here that look pretty sad. So the disease is present, the plants are really struggling. And then this pie chart with the different colors is trying to get at what's your diversity of the microbes in that soil system. And you can see It's modest. It's low. And then they say, okay, like with that story I was telling you with the potato breeders, they couldn't get disease to show up on their cultivars anymore. So what happened in that soil system over time is it was a conducive field to potato scab disease, and then it became suppressive over time. And when we went in to look at what's going on in that soil system, if you grew potato plants, if we pretend these are potato plants, they grew just fine. And when you dug up the potatoes, they weren't diseased. And so now we say it's a suppressive soil for potato scab disease in this case. And when we sampled the soil and started looking at what's going on with the microbial composition, we saw that there was more diversity. And in particular, when with the potato scab soil, this isn't the potato scab soil, this is a different system, but trying to use that story. With the potato scab soil, we found that there was actually really high populations of Streptomyces bacteria, a very particular type of bacteria called Streptomyces. That's the genus name. And that there were many different species of those in the soil system. So what they're showing here is that on this graph, they have time on the x-axis and then disease severity on the y-axis. And then you can see the pathogen is gaining entry into the field. And you can see that the disease is pretty bad. You know, the plants really get attacked. They're very susceptible. And you get a severe disease outbreak. Then you can see this downward trend in disease severity. So what they're trying to show is if you look up top here, they're trying to say that's when you start to get a shift in the microbe composition is during this downslide here, where you're starting, something's going on in the soil system that's causing the disease severity to go down. And then if you get into a true suppressive soil system, then if it does convert to a suppressive soil system, if that pathogen gains entry again to that field, the same pathogen, not a different one, the same species like the potato scab pathogen, and those plants get exposed to that, you can see that the disease severity is very, very mild. It should go way up. That dotted line is what should happen, but you can see it was very modest in this last part of the line graph in the sense that because the soil has become suppressive to the pathogen, when those plants get exposed to that second introduction, they're like, yeah, whatever, I'm okay. So this is a thing. There's several natural suppressive soils around the world. Probably the most famous one is out in California. It's a wheat field. And then the one I studied with potato scab disease was in Minnesota. And so those soils are very, very good to study because it helps show, what changed. And of course, there's nearby conducive soils that you can use as a comparison to see what's going on. We looked at all kinds of things about the soil, like what if the pH shift? What if the nutrient availability shifted? I mean, we looked at all the other like physical chemical characteristics of the soil, and there wasn't any significant difference with that. It was the microbes. Something changed. And so this idea of the soil legacy and the soil immune response, over time, you can get this shift from conducive to suppressive. So then when my postdoc advisor started looking at that suppressive field, she's like, well, if we could study how this happened, like what's going on here, maybe we can artificially create a suppressive soil anywhere we want if we can figure this out. And so that's why we started studying that soil system to see if we could figure out a way to create a suppressive soil. And potato scab disease is like the fourth most important the fourth most important pathogen um, in when they rank out because it's found everywhere around the world. Anywhere you look, you'll find this pathogen in soil systems that can handle potato growth. Here's another visual trying to get at this idea of soils that become, that are conducive to the disease, the initial outbreak, could see, at first there's just a few of the red plants showing up, the diseased plants in that monoculture. But then you can see phase 2 is very severe. And then how you start to get the suppression. And the line graph below it is just trying to match up to those figures of the field, showing how you get the decrease in disease and the increase in suppressiveness. So when we were looking at this idea with the suppressive soil in Minnesota, we noted that there were a lot of different Streptomyces bacteria that could inhibit the pathogen. You are all familiar with Streptomyces bacteria, even if you don't know it. If you've taken an antibiotic, most of the antibiotics in production today are actually still made by Streptomyces bacteria. They grow them up, they get them to make the antibiotic, and then they extract and purify it and give it to you when you're sick. And so the ability of them, they're phenomenal at producing antibiotics, all kinds of different antibiotics. And so that's why we zoomed in on them, because they were very present at very high levels in this suppressive soil. And we knew they produced tons of different antibiotics. So we were like, and sure enough, so many of the Streptomyces bacteria that we isolated and tested against the pathogen were killing the pathogen in coplate studies in the lab. So then we took it to the field, right? The next step is, you screen them in the lab, you're trying to figure out who can kill the pathogen. And then if you find one that's doing a really good job, then you go ahead and grow it up to high levels and you go to the field. And so then we took it to the field and did field studies where the farmer would, create the rows with his tractor. And then we had to go in and hand plant all the potatoes. So we would put the biocontrol Streptomyces into the soil, put the tuber down, close it up, and do that on and on and on. Wait for the potatoes to grow, potato plants to grow, get potatoes at the end of the season, and then we'd rate their disease severity, which you can just look at the surface of the tuber to rate the disease. And we got good results the first year. It was great. It's a statistically significant decrease in disease. The next year, not so good. And we started to realize putting one biocontrol agent into the soil might not be the best idea, right? This idea of, cocktails, a mixture of microbes, going back to the other picture. Maybe it's better to have more than one biocontrol agent. And so having a mixture instead of just one biocontrol agent. And so that's where some of my work, I was very interested in looking at, okay, how do we create a mixture of biocontrol agents? We don't want them to kill each other. That would be futile. So you want microbes that can kill the pathogen, but they don't kill each other. And they actually work together as a team to really synergistically kill off the pathogen. And so I was very interested in looking at that. And once we started to do 2 or 3 different microbes into the soil system, we got much better control of the disease. So good that Frito-Lay came knocking and said, hey, could you give us some of your microbes and see if we can. So they gave us some of their fields. The Frito-Lay had, thousands of acres in production, and they gave us some of their fields to play around in. And we did that year, we only saw a 10% reduction in disease. So we're all really disappointed. We're like, only 10% reduction. Because in the past, we've had like 40% reduction, 50%, even better than that. And so we were pretty disappointed. They were thrilled because they said, when you consider the thousands of acres we have in potato production, 10% is a good thing to think about. So they were fine with that. So that was kind of an interesting little study that we did with them. But they are thinking about, okay, can we use mixtures of microbes? And instead of one microbe at a time, when we're trying to come up with biocontrol agents, can we do, a synthetic community of biocontrol agents where we create mixtures? And so that's a big thing right now. There's a big push for that. And that's where they started using the word syncoms. So that's where that's coming from. It's human-made decisions on what microbes are going to go together. And then they said, or, another tactic we could take is use the microbes that are there, native to the soil ecosystem, and really look at them. And that's kind of what we were doing too, because we went to the disease suppressive soil and said, who's there? What are they doing? And can we use them? And so we were kind of doing both well, all of this really when we were getting into how can we control disease. Another idea, so this integrated pest management idea is breeding, taking your plants and breeding them for genotypes that are really good at either recruiting good microbes or they themselves are able to better defend themselves against the pathogens. And then another idea that they're getting into is the augmenting of soil systems with prebiotics, essentially, where you learn about the microbes, like the beneficial ones, the biocontrol agents, and you try to see what makes them happy in terms of what do they need to survive in terms of nutrition. Can you feed that into the soil to feed them so that their population levels stay high? And then they're able to do the job of killing off the pathogen that's trying to attack the plant. So all of this, there's lots of different angles that are going into this now in terms of how do we use microbes for um, disease suppression, and can we create suppressive soils through this, several ways of looking at it and using different tactics. So this is all very, very active because there are bans going into play on certain pesticides and fungicides, and so they have to come up with other strategies. And so this has really kind of kicked it into high gear when the banning of these pesticides started to go into effect. And so all of a sudden, biocontrol is a thing, whereas before it was very much fringe science. When I was actively involved in it in grad school, it was postdoc, it was like fringe. It's like, oh, we don't need to worry about that. We'll just use this chemical. It's all good. So they have to study what's going on for these microbes in the soil, the plants, how they're impacting that. This is a study that was nicely done for looking at what happens. So take a step back. Potato scab disease is a soil-based pathogen. It lives in the soil. It attacks the tubers that are in the soil. Now we're going to flip this a little bit. This is a pathogen that's above ground. All right. It attacks the leaves. It's a Pseudomonas species. And what was surprising to find is how the root system, even though it wasn't the roots area that was getting attacked, the signal was coming from the leaves down into the roots and essentially impacting the microbes. Hey, I'm getting attacked here. And so this study was very nice to show how you can have that above ground pathogen infection and it can still influence what's happening in the soil system near the roots. And so Pseudomonas syringae, Papaver tomato, is the pathogen that they were working with. And what they did was they grew here we conditioned soil by growing multiple generations, so 5 generations of Arabidopsis, so that's the guinea pig plant, and they inoculated it on purpose with Pseudomonas. So 5 times of doing that in this pot of soil or these pots of soil. And then they looked at the microbes that were there, the rhizome here. They looked at plant performance in a 6th round of Arabidopsis. Right? And the 6th one they didn't infect. And they just grew Arabidopsis a 6th time to see what would happen. And then they looked at the root exudation profiles as well, and looked at microbiome, what's going on with that in the soil. And so this has led to a hypothesis that's out there now called the cry for help hypothesis. I would like you to know that phrase, know what I'm talking about when I say that. But basically it's the plant saying, eek, I'm getting attacked. Hey, microbes, can you help me out? And so this is the idea that when a plant is getting attacked, that they will be shifting their root exudation patterns and ratios in order to recruit the microbes that should hopefully help them. So they're crying for help by shifting their exudates to try to recruit the right bacteria or fungi that can help them with getting attacked. In this case, getting attacked by Pseudomonas. So they see that both with, we're talking about a biotic thing where it's a bacterial pathogen attacking, but they are starting to see abiotic stresses too. This cry for help can happen under those conditions too. So some of the data that they were collecting, so this is going back to the growth defense trade-off, where they were saying when you have pathogen conditioned soil, so those 5 generations of Arabidopsis that were infected with Pseudomonas, that's your conditioned soil, your pathogen conditioned soil, and then just your control plants that were grown without the pathogen. And then the 6th generation of plants was planted on these soils. And that's what you're seeing. So they're trying to figure out, how did the plants grow the 6th generation? How did they grow into those soils, of those conditioned soils? And you can see they actually didn't grow so well in the pathogen conditioned soil. But it kind of makes sense because that's the whole growth defense trade-off. So if these Arabidopsis got plunked into conditioned soil where the 5 generations of plants had been infected, and when growing in that soil, they were getting the signal there's pathogens around, and so they weren't investing in growth. And that actually supports this idea that you get that shift into, well, I'm going to defend myself. So it's essentially like the Arabidopsis plant that got stuck into the pathogen conditioned soil. It could tell by the mixture of microbes and what was going on. Oh, it seems like there's a pathogen around, even though there really wasn't, but it kind of got faked into, I'm going to try to defend myself. Whereas the control, they were growing quite fine. So that was an interesting result. Or, oh, I was talking about the wrong one. Biomass. This is the one I was just talking about. It'd help if I wear my glasses. So here's the biomass, sorry. So here I was thinking biomass when this one was showing disease incidence. So let me go to this one. I apologize. This is the one I was just talking about. The 6th generation of plants, or the 6th um, set of plants that got put into there. You can see that when they were in the pathogen conditioned soil, they were not growing as much. They were activating their defenses, thinking that they were going to be attacked because the microbial community in that pathogen conditioned soil was basically giving them the signals that there's pathogens around. And then here, when they were just put in the control soil, they grew fine. This one, on the other hand, is disease. So here we're looking at percent disease on the y-axis, and you can see that big difference, significant difference in terms of disease. When that 6th group of Arabidopsis got put into those conditioned soils, the control that didn't have, no Pseudomonas was involved in those plants at all. You can see they grew just fine, or they had high disease incidence because they were in a soil system that was not conditioned by plants growing with the Pseudomonas. So now they're at risk. Whereas the plants that got put in with the pathogen, when the pathogen was present on those plants, they got put into those soils and everything went fine. The disease incidence was much lower. So again, pointing to the idea that there is this soil-borne legacy, the shift in the microbiome because of the pathogen presence in those previous generations of Arabidopsis. You do see a difference in disease incidence compared to the control. They also were seeing a difference in hormone shifts in the plant. So this is when the pathogen was present on Arabidopsis. That's the blue bars. So that soil system, when they were looking at phytohormones, you can see there's kind of a either or. So JA is jasmonic acid. So that's one hormone that was really quite upregulated when the pathogen was present. So jasmonic acid was helping the plant deal with this bacterial attack, whereas their salicylic acid was very modest. And so they're showing that the hormones, different hormones come into play when these plants are getting attacked by, in this case, it was a bacterial pathogen. So it had the pattern of ramping up jasmonic acid and kind of shifting down salicylic. And abscisic acid didn't really do a whole lot. Then when they were looking at differences, they didn't really see much. Okay. So it triggered the plant growing in that pathogen conditioned soil. When that sixth group of Arabidopsis got put into that pathogen conditioned soil, it quite dramatically triggered those plants to go ahead and start synthesizing jasmonic acid to defend themselves. So it was like the microbes were saying, hey, you're going to get attacked by, Pseudomonas, you better ramp up. And they did. They kicked up on their jasmonic acid to defend themselves. And then here they were looking at root exudation patterns. And I'm not going to get into this in huge detail. You can see on the x-axis that they were looking for many different types of root exudates. Big picture idea here is that the bars aren't identical to each other. They're red and blue. They're different. And so you definitely get a different profile of different types and ratios of these root exudates, depending on if the plants were exposed to Pseudomonas or not, and then what was happening with the root exudation pattern. So clearly they can show, the conditioned soil from the pathogen being present versus the control. You definitely have a shift in exudation patterns. So that was a really nice addition to this study. So 5 generations of plants that were infected with Pseudomonas did shift and recruit the microbiome distinct from that control that didn't have the pathogen present. I didn't get into it, but you could say, well, it's just the soil. It's not the microbes in the soil. It's just the exudates in the soil that are doing all the work here. But they did an experiment where they got rid of the bacteria in the soil, and you could see it fell apart. And so they were able to show the presence and shift in the bacteria is what is the difference here. It's not a difference in the chemical composition of the soil. It's the microbial composition of the soil. So here in a foliar pathogen, so it's attacking the leaves, but what happened was it caused a shift in the root exudation pattern that impacted the microbes in the soil. They became altered because of it. And so they're saying, yep, it's a soil-borne legacy that allowed for the plant to resist disease in these that last generation of plants they put into there. So really nice study. Here's their graphic where they're trying to show this in a visual way. So plant systemic signaling leads to a change in root exudation profiles, which in turn shifts the microbiome. It helps induce resistance against Pseudomonas for the future plants that come in. So our results show that above ground infection by Pseudomonas really does affect the resistance of subsequent plant populations in that soil. And it's due to the mechanism of what's happening in the soil system, that soil-borne legacy. So really nice study showing that you can have above ground interactions going on that can profoundly influence what's going on in the soil and shifting the microbiome there. Different microbes are showing up to then help the plant defend itself against this particular Pseudomonas pathogen. So lots of research is going into this kind of idea where they're looking at how does the microbiome shift in the presence? And when we see good disease control, let's figure out what's going on with the microbiome because we think it's the microbiome driving this. It's not really just, something chemical going on in the rhizosphere. We have to have the microbiome mixture to be the right one to help the plant fight it off. But then when you get into that, you also have to think about, okay, if we have these microbes and we know this group of microbes are really good at helping the plant defend against Pseudomonas, but then you have to think about if you want to use those in different soil systems, you have to make sure that they can survive in those different soil systems. You have to make sure they're active in those soil systems, that they persist in those soil systems, and that they don't just form spores and go dormant and take a long nap. And so there's a lot to it, but there's been a lot more research done in the recent past here on the microbiome and a nice, diverse, healthy microbiome to help the plant against disease, which is huge. Okay, so the next topic I want to get into is a study that my postdoc advisor did. So Linda Kinkle is my postdoc advisor, and she did a nice study looking at using streptomyces in the soil as biocontrol agents, but she was manipulating plants again, kind of similar to what we just looked at, but in this case, she was looking at what happens if you have a monoculture of plants versus more diverse plant groupings. What happens below ground? And she was in particular looking at the Streptomyces bacteria for their ability to be very inhibitory. So we know that Streptomyces bacteria are good biocontrol agents because they release a lot of antibiotics that can kill pathogens. And so she was zooming in on that. How do we get like souped up Streptomyces bacteria that can really kick and kill pathogens? And so she was looking at how should we manage the plants and the associated microbes to do this. But that's going to take a while. So we're going to stop here because it's a story I got to tell, and it's going to take a while. All right, so we'll stop there. So today's Wednesday already. Monday, we will start getting into come prepared with your laptops. We will start, part of the class time will be for looking for papers that you want to work on for a group presentation. Okay, so you're going to be doing a little bit of searching with Google Scholar or something on Monday.